So I wanted to give a quick introduction first to Regeneron. If you have heard of us before, you've probably heard of us from our antibody programs and our other biologics. But really leading from our mission, our mission is to repeatedly bring important new medicines to patients with serious diseases, and that's really modality independent. So we've had some really tremendous innovations with biologics like traps and antibodies, and now we're really pushing into gene therapy because we think that there's incredible potential there. So I'm going to start actually with CRISPR-Cas9, which is the basis of the gene therapy that I'm going to talk about today. So CRISPR-Cas9 is a system to really exquisitely edit the genome in a sequence-directed way, so at a very specific location in the genome. And the technology is only a little over 10 years old, but already there's been a number of applications. It's widely used in preclinical research and agricultural applications, in diagnostics, and in the clinic in ex vivo applications. And today I'm going to talk about the in vivo applications that we're pioneering. So Regeneron, along with our partners Intellia, are trying to develop CRISPR-Cas9 approaches for gene editing in vivo. So in vivo being separately, so in contrast to the ex vivo approaches that we just heard about, where cells are removed from the body and then edited and then put back into the body, this would be a therapy that you would potentially receive via an infusion. So there's two different types of applications that we can conceive of with CRISPR-Cas9. So one is a knockout, which isn't really relevant to the Pompe community, but I bring it up because this is a therapy that's already in the clinic. It's in phase 3 clinical trials, and this is the first proof of concept for gene editing in vivo in a human. We also have been working on technology to do the gene replacement, as Nina described it. And I'll go into more detail about how this works, but we're pursuing this for multiple therapeutic proteins. The most advanced program we have is for hemophilia B, and that clinical trial, it's just opened, it's currently enrolling. So I'm going to show you a little bit of data from the factor IX studies to demonstrate how we can use this approach to express a therapeutic protein. So the goal here is the approach that we're taking is to try and edit a small number of hepatocytes in the liver to act as an in vivo biofactory. So this is a similar approach to some other gene therapies that use an AAV episome to recruit cells in the liver to express a therapeutic protein. But this approach is a little bit different because instead of using the AAV that exists as a separate piece of DNA, we're actually putting the gene directly into the genome. So we've chosen the albumin locus. This is one of the most highly expressed genes in the genome. So we can modify a small percentage of these alleles. It doesn't affect albumin levels, at least in the animals so far. And but it really is very potent for expressing a lot of protein. So we have two components. We have up top, you can see the LNP, and this is what delivers the CRISPR-Cas9 system to make that cut at that specific region in the genome. And then separately, we use an AAV to bring in the piece of DNA that's going to get inserted. So when the cell goes to repair that cut, that's when it will actually insert that piece of DNA, that being present there at the right time if we co-administer these. So we put this in between two exons, the first and second exon of the albumin locus, and our AAV, so because we use that albumin locus, it doesn't have to have a promoter. So that's a safety consideration. If this accidentally goes somewhere else in the genome, there's nothing there to drive gene expression, so it's less likely to affect any upstream or downstream gene that it might land in. So that's the idea. The albumin and that small percentage of loci and that small percentage of hepatocytes is driving the therapeutic protein. So this is data from our factor IX preclinical work. And I put this slide up to demonstrate that we can titrate the dose to get different levels of expression. We can titrate either the amount of the CRISPR-Cas9 system we put in, or we can titrate the amount of the template and try to hit a sweet spot. In the case of a therapeutic protein like factor IX, we really don't want to overexpress. But in the case of GAA, we really want to get as much as we can safely in there. So What I really want to emphasize is what we see in preclinical, at least, is the durability of this approach. So as I mentioned, conventional AAV exists as episomes, and in a rapidly dividing tissue, these episomes can be lost. And that's what we see here in the left is showing when we treat neonatal mice on the day that they're born, mice that receive a conventional liver-targeted episomal gene therapy rapidly lose expression, whereas mice that were treated with gene insertion maintain that expression as they develop. I want to just, since we have a little bit of time, just to point out that the liver here is a rapidly dividing tissue. It's a little bit different than the muscle-directed therapies that we heard earlier. The muscle develops a little bit differently, and we can go through that in the question and answer if we want to. So I also want to point out on the right is a very extreme experiment where we take animals, treat them, and then remove 2/3 of the liver. The liver has an amazing regenerative capacity. In 1 week, it will regrow to its normal size. So it's a very extreme model of liver damage. So you can see in mice that were treated with the conventional episomal gene therapy, after this regrowth, the expression is lost. But even with this extreme model, in mice that receive the gene insertion, because it's going into the genome, and that genome is being replicated, we maintain expression. I'm sorry, excuse me. Okay, so now I want to switch to Pompe disease and talk about our molecule. So as Brian and Russell really elegantly pointed out earlier, GAA has difficulty getting from the bloodstream into tissues. So we didn't want to come in with a regular GAA knowing that we were going to have that challenge. So we have been working on using our antibody platform to try and improve GAA, to improve its ability to get out of the bloodstream, to get into tissues, and to get into lysosomes. And the way that we've been doing that is by using small antibody fragments. So it's just the very tip of an antibody that binds to another protein and recognizes that protein. We take just that small piece and put some linkers in and then attach that to GAA. So GAA gets processed when it gets into the lysosome, and this naturally gets cleaved off. But while it's circulating, these scFvs can make a big difference in where GAA gets distributed to. So we've been working on a couple of different receptors and a lot of different antibodies to find the optimal way to do this. So as was mentioned before, GAA, the average GAA uses mannose-6-phosphate to get into cells. So our scFvs use a similar kind of idea. We can just pick the receptor that we want to use. We're not restricted to mannose-6-phosphate. So show one example here from a publication where we picked a receptor that's highly expressed on muscle and compare, so in the top two panels are a wild-type mouse and then a Pompe disease model mouse, and all that dark purple staining is glycogen. The lower two panels is a mouse that was treated with an unmodified GAA and then a mouse that was treated with the targeted GAA. There's a really dramatic difference in glycogen clearance. But we can expand this, and we really want to get into the brain as well, so we can treat potentially all of the phenotypes of Pompe disease. So we've also been working on a transferrin receptor targeted version of this. And I'm showing there a graph of glycogen in the brain of a mouse that was treated with this transferrin receptor GAA. And you can see that we're really eliminating the extra glycogen in multiple regions of the brain, and the spinal cord. Okay, so when we put all these things together, the gene insertion and the targeted GAA, what do we get? So here I'm showing mice that received gene insertion, and we looked at them 3 weeks after we dosed with gene insertion. Looked at glycogen in multiple tissues. I'm showing here the heart, the diaphragm, the quadriceps, and we're really eliminating most of that extra stored glycogen. And in the brain, here's where you can really see a difference between that targeting. So the transferrin receptor GAA is getting into the brain, whereas the CD63 GAA it's a partial reduction. It's not really getting across the blood-brain barrier like the transferrin receptor GAA. So I'm going to continue with the transferrin receptor GAA. I want to show you functionally what this means for mice that are modeling Pompe disease. So these mice we treated as, we say young adult mice, 2 to 3 months old, and then followed them for an additional year. So as you can see, the body weight in the treated mice in pink matches very well, tracks very well with the wild type mice. Functionally, we can see a significant improvement in tests of motor coordination and strength. So looking at the wire hang and balance beam at the end of the study. So in the wild type mice, you can see there's an age-related decline in strength in the wire hang test. We see that also in the treated and the untreated mice, but we do see that significant difference that's maintained throughout the course of the experiment. So the last thing I want to show you is some data from non-human primates. So these aren't a disease model of Pompe disease, but we can ask, can we translate this model to primates? So we are able to do the gene insertion in cynomolgus monkeys. We can see an increase in the amount of the circulating targeted GAA. And then if we take tissues and look specifically for the lysosomal fraction of GAA by using a Western blot, looking for that cleaved GAA, we can see an increase in the amount of GAA over the wild type levels in heart, diaphragm, and quadriceps. And for the transferrin receptor targeted GAA, we can also see it in the brain. Okay. So in summary, we're really excited about this program. We're trying very hard to get it to the clinic as soon as possible. So I showed you a little bit of background on the CRISPR technology, the knockout program for TTR, which is currently in phase 3, the Factor IX program, which is currently enrolling for phase 1, and then how we can apply this potentially to GAA. And particularly using those targeted transgenes that we think really is able to get more GAA into the tissues that need to be treated. So this is my final slide, so thank you very much.